Osteoarthritis affects 595 million people globally. Despite the pain and physical limitations it causes, there are no disease-modifying drugs available to stop its progression. The difficulty in treating it lies in its timeline. The physical degradation of the joint is a slow burn that takes decades to manifest. A potential solution comes from an existing treatment, Metformin. It is a widely used, inexpensive oral medication typically prescribed for type 2 diabetes. Preclinical studies indicate metformin may offer chondroprotection. The drug appears to trigger specific cellular pathways, namely AMPK and GDF15, protecting cartilage and reducing joint inflammation. The medical community might already possess a chemical tool to slow or halt cartilage degradation. The primary barrier to proving it is not biological but logistical. To prove a drug's efficacy, researchers rely on the randomized controlled trial. These trials typically track patients over a span of 1 to 3 years. Osteoarthritis develops over 20 to 30 years. A standard trial captures only a fraction of the disease's life cycle. Running a controlled trial for a decade, monitoring thousands of patients to see if a preventative drug stops a slow-moving joint disease, requires prohibitive amounts of funding and continuous patient compliance. To study long-term effects, researchers turn to retrospective observational cohort data. They look backward through decades of patient medical histories. However, these datasets contain inherent flaws. They are heavily compromised by genetic confounding and indication bias, meaning the underlying health conditions that caused a doctor to prescribe the drug in the first place skew the results. Researchers face a methodological deadlock. Randomized controlled trials for osteoarthritis prevention are practically impossible, and standard observational data is too noisy to isolate true causation. To cut through this observational noise, a team of researchers utilized the Swedish Twin Registry to conduct a co-twin control study. The baseline biology of twins provides a natural control. Monozygotic or identical twins share 100% of their segregating genes. Dizygotic or fraternal twins share roughly 50% of their genes. Importantly, both identical and fraternal twin pairs share the exact same early familial environment. The study relies on the concept of the discordant pair. This occurs when one twin initiates treatment with metformin and their genetically related sibling does not. By measuring the difference in outcomes within these specific pairs, the researchers mathematically neutralize the unobserved genetic and shared environmental variables that typically ruin standard observational studies. The genetic relatedness between the siblings creates a natural experiment. It closely mimics the random assignment of an otherwise impossible 10-year clinical trial. This flowchart details the data selection process. From 109,000 twins, researchers filtered for eligible individuals aged 35 to 75. They systematically excluded pairs with pre-existing osteoarthritis or prior metformin use. This distilled the cohort to exactly 1,261 discordant twin pairs, differing entirely by their treatment status. Through aggressive pruning of the database, the researchers converted chaotic population records into a strictly controlled scientific test arena. Even within twin pairs sharing genetics and early environment, adult lifestyles diverge. Individual-level variables still exist that can skew the results. In this study, body mass index represents the most significant confounding variable. A high BMI presents a dual threat. It independently increases the risk of developing diabetes, which necessitates the metformin prescription, and it acts as a constant mechanical stressor that physically degrades the joints, causing osteoarthritis. To verify their methodology, the researchers ran a negative control. They tracked random joint dislocations, events entirely unrelated to diabetes. Both the treated and untreated twins experienced dislocations at similar rates, proving there was no hidden bias in how often the different groups sought medical care. The researchers mathematically adjusted the final model for individual differences in BMI, smoking history, and education. This final adjustment isolated the specific effect of metformin on joint cartilage. Twins who initiated metformin treatment demonstrated a significantly decreased risk of being diagnosed with incident osteoarthritis over the 11-year follow-up period. This table breaks down the hazard ratios. Looking at the adjusted stratified Cox regression, the treated twins showed a hazard ratio of 0.60 compared to their non-treated co-twins. To see how this ratio translates to actual disease progression, we look at the standardized survival function in Figure 2. Starting at the exact same point, the survival curves visually diverge, the protective effect compounding, separating the treated line year after year. By the 10-year mark, 6.7% of the treated twins had developed osteoarthritis, compared to 9.7% of the untreated twins. The data establishes an absolute risk reduction of roughly 3 percentage points over 10 years. In the context of joint survival, this represents a measurable, sustained delay in physical degradation. An absolute reduction of 3% may sound small in isolation, but we must evaluate it through the lens of the population attributable fraction. When you apply a 3% absolute drop to a global population of hundreds of millions of people at risk for joint disease, it translates to millions of prevented cases and a massive reduction in the worldwide healthcare burden. However, the study carries a critical limitation. The researchers relied on specialist care records and lacked access to primary care data. This means they could not adjust for the baseline severity of the patient's underlying diabetes. Clinically, a doctor cannot ethically prescribe a heavy-duty diabetes medication to healthy individuals in the general population merely to protect their cartilage. Despite these clinical barriers, the twin design methodology holds up. The researchers successfully bypassed an unfeasible 10-year clinical trial to extract a genuine causal insight from observational data. While metformin is not a blanket preventative prescription for joint pain today, this study validates the biological pathways it targets. It establishes those cellular mechanisms as the primary candidates for the future of osteoarthritis disease modification.